Welcome to cryotalk, a bite-sized bio podcast sponsored by Thermo Fisher Scientific, featuring conversations between your host, Ava Amson, and experts in the field of cryo-electron microscopy. Today on cryotalk.
Today on cryotalk, we're joined by Peter Shen, Assistant Professor of Biochemistry at the University of Utah. He tells us what initially drew him to cryo-EM.
Way to answer mechanistic questions about biology is to directly visualize them. In fact, in my lab, we have this theme, or a motto, if you will, seeing is believing.
How creating the Cryo-EM 101 course led to developing merit badges for the National Centers for Cryo-EM.
It was during the middle of this funding period where we realized that we need to continue to innovate and develop resources to bridge that expertise gap.
And what he does in his spare time.
We have a weekly series of pickup basketball that's led by graduate students and postdocs, and they've been generous enough to let old faculty members like myself join them.
All in this episode of cryotalk. Hi, and welcome to cryotalk. I'm Ava Amson, and I'm here today with Peter Shen. Peter is Assistant Professor of Biochemistry at the University of Utah. So hi, Peter. How are you?
I'm great. Thanks for asking, Ava. I'm glad to be here.
Yeah, great to have you. Now, the first question that I usually ask people on this podcast is if you can just maybe tell us a little bit about your career so far.
Yeah, happy to. So I'm an assistant professor here at the University of Utah, and I've started my lab in 2017. So I've been running my lab for about 5.5 years now. And the theme of my lab is that we care about the mechanisms underlying protein homeostasis. And this really refers to the balance between how cells make proteins and gets rid of proteins that either the cells don't need anymore, or perhaps problematic proteins that could cause harm to the cell. And so there's the constant balance between how cells make decisions of how when to make proteins or how to make proteins, and how to get rid of proteins that are no longer needed. And so in our lab, we have a lot of projects that cover different aspects of this theme, including how proteins are made at the ribosome, how proteins become folded, and also the flip side of that equation in terms of how proteins become unfolded prior to their degradation.
It sounds like it's very much like the basic principles of biochemistry that you're trying to figure out.
Exactly, yeah, and I think that's really what has drawn me to this field, is that it is very fundamental in nature. All of our cells rely on the orchestration of proteins doing their fulfilling their intended functions. And unsurprisingly, there are many diseases that are associated with proteins not being able to work properly. So this is a theme that has attracted me for a long time, ever since my postdoc, and so excited to be able to run a research program to be studying this very thing.
And one of the tools you're using is cryo-EM. So how are you using that, and how did you get started with cryo-EM?
So I'll answer the second part of that question, how I got started first. I like to say that I was doing cryo-EM long before it was cool to do cryo-EM, no pun intended. But I became first exposed to cryo-EM when I just started graduate school at Brigham Young University in 2006. And at the time, I had never heard of the method. Certainly, the field wasn't where it is now in terms of technology or capabilities. And we were fortunate at the time. The university had a great microscope, a 300 kV FEI TF30 microscope, side entry holder. And at the time there was a lab. I ended up joining the lab of David Belknap to study virus structures. And so cryo-EM at the time was great for studying things that are really big, things with a lot of internal symmetry, which viruses certainly fit the bill. And the reason I got drawn to that is because, I remember the very first time I saw a cryo-EM image, and I just couldn't believe that we could see these biological particles with our own eyes. I remember the first time seeing a reconstruction and thinking, oh, is this a simulation? But no, these are actual reconstructions generated from cryo-EM images. And of course, the field at the time, was very limited in terms of resolution, but that's how I became initially exposed to the field. And then, over the years, I like to think that I'm just so lucky to have been in the right place at the right time, to have stayed in the field and to have witnessed the amazing technological advances, and thinking that the best way to answer mechanistic questions about biology is to directly visualize them. In fact, in my lab, we have this theme, or a motto, if you will, that seeing is believing, that there really is no better way to understand biology than to be able to directly visualize the protein complexes that we're interested in. And so I've come a long ways from studying virus structures to now studying still big complexes, but much smaller than viruses, and through cryo-EM.
Yeah. So have you noticed that over the years, obviously the technology has changed with you as well? Are you taking advantage of the technological advances in cryo-EM?
Yeah, I am. I think, the field continues to be in a golden age. It's still so exciting to see the advances that continue to be made in terms of hardware advances and software advances, where we're not only being able to improve resolution, but also to recover multiple conformational states of a protein or a target of interest. And so my lab, we try to stay at the forefront of the technology. Just this morning, I was playing around with this new tool from Shore's Shears Group that has an automated model building feature, where if you have a good enough 3D reconstruction, it can automatically build an atomic model for you.
Oh, wow.
And yeah, it's a fascinating tool. And so I think the direction of the field is that, people are still innovating, and that we're really not, we're really just scratching the surface in terms of the potential of what we can do with cryo-EM and image processing.
Yeah. Yeah, and I love that motto, seeing is believing. That's definitely what it's all about with cryo-EM.
Yeah, I mean, just to add to that, I remember my first experiences, doing biochemical experiments as an undergraduate student or in undergraduate labs, and thinking it was really cool that we could see bands on a gel for proteins and DNA, or can see spectral peaks for different spectroscopic techniques. But it's always satisfying to me to be able to actually see the thing that we're studying.
Yeah, it's a different level.
Um,
Yeah, and now you've also been involved with cryo-EM training, for example, with the National Centers for Cryo-EM. Can you tell us a bit more about that?
Yeah, I'm happy to. So so I'm part of a program that's funded by the NIH that has led to the creation of what we call cryo-EM 101. And this was created in partnership initially with Janet Iwasa. And over the past couple of years, we've also extended or expanded our program to also create training for cryo-electron tomography, cryo-ET 101. And this is in partnership with Julia Brash. And so the history of this is that in 2017, this was around the time when the NIH started to put out calls for proposals, grant applications to create curriculum development materials, recognizing that there is quite an expertise gap in the field, where there's burgeoning interest in people wanting to learn cryo-EM, and the NIH was ready to fund national centers towards this so-called synchrotron model, such that not every institution is going to have their own cryo-EM facility. And so there, how do we bridge that gap between those who don't have experience doing cryo-EM but want to gain access to national centers? And so at the time when we applied for this curriculum development grant, I don't think that we intentionally could foresee that we would be working so closely with the national centers. But in hindsight, of course, this is what needed to happen. And so at the time, Janet and I, we created cryo-EM 101, which covers the basics of really all practical steps of a cryo-EM workflow, from sample purification and grid preparation all the way to data collection and data processing. But at the same time, these national centers were up and running, multiple high-end, top-of-the-line microscopes at each facility. And they would also provide their own training courses. And we're glad that they're also using a cryo-EM 101 material, but there still is that skill gap between those who don't have the exposure to cryo-EM resources, and how do people know if their sample is good enough for cryo-EM? And so it was during the middle of this funding period where we realized that we need to continue to innovate and develop resources to bridge that expertise gap. And so this has now led to the creation of what the national centers leading this so-called merit badge program, where students, learners can earn specific merit badges that cover a distinct aspect of the cryo-EM project workflow. So for example, how do you freeze a grid? And so this provides some level of certification so that newcomers can essentially be certified to use these instruments. And the spirit of these merit badges is then that they're cross-honored across the three national centers, NCCAT, PNCC, and S2C2 at Stanford.
So I guess that standardizes the bit, so now they all have access to the same information.
Yeah, that's a great way of putting it. It standardizes the procedure, and and this is something I think the field needs. And especially if you have a trainee and you're going to put them in front of an expensive instrument, you want to have a certain level of trust that you have them freeze their grid, but that they know what they're doing. And so having that certification through a merit badge would add that confidence. And so I think the spirit of the program is exactly what the field needs.
And do you have any insight in who has used the cryo-EM 101 resource since it launched? Like how many people, where are they coming from?
So we recently reported this to the NIH, so it's a little bit fresh in my mind. But last I checked, which was a few weeks ago, we were at about 43,000 unique learners worldwide, and about half of them come from the United States. And perhaps not surprisingly, the hotspots of those who are visiting cryo-EM 101 or using cryo 101 material are those who do have existing cryo-EM infrastructure, but probably have a growing base of trained, a growing base of newcomers who want to know the very basics. And so the way the cryo-EM development groups have been structured, I think has been really nice, where we have different levels of expertise and also different groups who cover different aspects of cryo-EM. So for example, we consider ourselves, we really thrive by the 101 level of Cryo-EM 101. I just really focus on the basics. But then if people really want more deeper knowledge, then there are Grant Jensen's, very well-known series of lecture videos. Fred Sigworth has a series of videos that really takes a deep dive. That's more very advanced cryo-EM theory. And then there are more hands-on curriculum development tools through using VR tools that are developed by a group at Purdue. And then more recently, is a group funded to develop more image processing-based tools called cryo-EDU, where they do simulations of image processing results so that people who might not necessarily have their own computational resources can at least go through these simulations and see what happens when they make good or suboptimal decisions and the effects of those decisions on their outcomes. And so it's been really neat to see how these curriculum developers have come together and offer complementary perspectives. And so here at with our cryo-EM 101, it really does focus on the basics. What does a good sample look like? What does a suboptimal sample look like? And once these newcomers have established, or at least have some basic understanding of calibrating their expectations, then I think this can save a lot of time and resources in the long run.
Yeah, definitely. So I guess you also have a little bit of insight in the variety of people that use cryo-EM. Do you think that has changed over the years? Like when you first started, was there a typical type of cryo-EM user, and is that different from what they are now?
To think a little bit about this question, we do have limited access to this data because when we look at our web traffic analytics, it doesn't break down the types of visitors, but we do have a way where people can provide feedback on course materials, or if they have more specific questions, they have been able to reach out to us specifically over the years. And my sense, and I don't have hard numbers to back this up, is that, but my sense is that the general proportion of training level has remained pretty consistent over the years in terms of most of the people who reach out to us are early stage graduate students or postdocs, but there certainly are a lot of faculty members that have also reached out. For example, trained crystallographers who are great biochemists and probably have great samples, but haven't had hands-on experience with cryo-EM instrumentation. So those would be faculty members who have reached out to us. But I would say for the most part, it's early stage graduate students. But the level of our training materials, I would say, even catered to undergraduates, people who are learning about structural biology at a very basic level, where nowadays, and I think as I harken back to my own undergraduate experience and learning about X-ray crystallography or NMR, certainly cryo-EM was not in that mix when I was a student 20 years ago. But now I think cryo-EM is probably part of mainstream undergraduate curriculum, and the way that we've designed cryo-EM 101 is that it should also cater to that audience as well.
Yeah. And do you think that having early hands-on experience with cryo-EM can help students find jobs and academic careers?
Yeah. So one of our biggest motivations for cryo-EM 101 is that someone can read all they want about cryo-EM theory, but there really is no substitute for hands-on experience. Now, that said, we also recognize that is a limitation of the training tools that we've posted on our website, because there is really no hands-on components. But we tried to emphasize, through the videos that we've posted, like walkthrough videos of, this is how someone handles a side entry holder. This is how someone clips a grid and prepare it so that they could be loaded into an auto loader. And so having those tools available at least helps the learners um, give them a certain level of preparation so that when it's time for them to have that hands-on experience, it's not a complete foreign experience to them. In fact, I would say that the ideal workflow is that a lot of the national centers, they host workshops for these purposes. And so prior to a student attending an in-person hands-on workshop, hosted by a national facility, for example, that they would have referred to Cryo-EM 101 materials that maximizes their preparation, again, so that it's not a complete foreign experience for them when they do it. But then it's, in order to be certified or to have that experience to be marketable for next steps, you have to have that hands-on experience. And I think there really is no substitute for just time in front of a microscope, or just that muscle memory of how do you freeze grids or how do you clip grids, the time in front of a computer screen evaluating what is a good image, what is a bad image. And so I think there are multiple layers, multiple steps to gain that expertise, but we're hoping that Cryo-EM 101 is a good starting point for that.
And do you think that over the years the instruments have become more user-friendly? Like, are there more entry-level instruments now for people to start using?
I definitely think that the field has progressed in the right direction in this regard.
So does that make it easier for students?
It does. And I think I would say in my experience, most students probably don't have as much time in front of the microscope or driving a microscope as they deserve. And depending on the facility, for example, it's understandable if you don't want to trust complete newbies to fiddle around with your multi-million-dollar instruments. But along those lines, though, it's the level of involvement to, for example, align a microscope has been greatly simplified, even to the point of automated, compared to 10 plus years ago. And so I would say that a lot of the skills that I learned as a trainee are probably considered antiquated, in terms of aligning a side entry microscope, or even operating a side entry holder. I think the fewer and fewer institutions are supporting those types of instruments, and seeing the new microscopes that are coming out where, for example, you have dedicated screening microscopes that are pretty automated. And so I think the level of, maybe not necessarily the level of expertise, but the focal points of the expertise have shifted over the years, where I don't know how many hours I've spent in, for example, developing film. Used to collect cryo-EM images on film, and no one does that anymore. And so for my own trainees, and I'm sensitive to this as I'm bringing in new graduate students and postdocs, that I'm thinking about, is it really worth your time? Would it make you more marketable if you had these skills? And maybe less so, and I would rather have them spend their time on using state-of-the-art instrumentation so that they can focus more on their own projects rather than the technical expertise.
That makes sense. Yeah, thanks. I want to change direction slightly and ask you, so we've talked a lot about work and cryo-EM, but what do you like to do when you're not working? Do you have any hobbies? Or
hobbies, some things that come to mind. I like playing basketball. And so our department, we have a weekly series of pickup basketball that's led by graduate students and postdocs, and they've been generous enough to let old faculty members like myself join them. It's hard to keep up with them, but that is one hobby. I'm at a stage of my life where a lot of my time is focused on the three kids that I have, ranging from elementary to middle school. And, that comes with a lot of time spent on sports and music lessons. But turning that into also a hobby where I'm practicing sports with them or we're playing music with them. And then and I would say another hobby that comes to mind, something that we've picked up over the past few years, is really enjoying the great outdoors here in Utah. Just have so much access to beautiful wilderness, including 5 national parks. In fact, just this past weekend, we spent the weekend down at a national park and got to get away from the city for a little bit.
Yeah, nice. When I heard you were in Utah, I was kind of expecting you to mention the outdoors, because it's one of those states where people go outside, and you live in a place where other people travel to go outdoors.
And so funny enough, I didn't go to my first national park until about 5 years ago. I've spent most of my life here in Utah, but I certainly didn't take advantage of the beauty that we have here until much more recently. And that was in part spurred by kids getting older and just needing to find more bonding activities. And boy, I just think about how much I've missed out on not taking advantage of that sooner.
Yeah. And when you're indoors, do you have any like things, like do you like reading or films or music? Any recommendations perhaps for our listeners?
When I'm indoors, at home we play a lot of board games. And it's fun where the kids are at the point where we could play more strategy-based games, and so we definitely.
Any favorite games?
It depends on who you're with. I would say if you're with a bigger group and you like chaos, one game that we love is called Captain Sonar. I don't know if you've heard of it, but you can think of it as a battleship on steroids, where you have 2 teams that is in charge of navigating a submarine, but trying to blow up the other submarine. And so the 2 teams are sitting on opposite sides of the table, and things are happening in real time. And so people are talking over one another, and you're trying to eavesdrop and keep track on what the other team is doing. And so that's a lot of fun. We like a lot of puzzle-based games, mixing and matching certain tiles. So the game Azul comes to mind. Another party game or go-to game that we like is called Code Names. But anyway, there's so many. So I think if you open our closet at home, there's probably more than 100 games, and some of them are probably still shrink wrapped. So that's something that our family definitely enjoys indoors. I'd say the other thing is that we like music, and so I play the piano. My wife plays the cello, and our kids play different instruments. We have someone learning the viola, someone playing the guitar, and then someone playing the violin. And so we like to find songs where we can play with each other or accompany one.
That's great. I actually play violin myself, so I love hearing that. And your kid that's learning viola is going to have a lot of options, because all the orchestras are always short of violas.
And that was a little bit of a tactical decision when he started to think about what instrument he wanted to pick up. And so, he's enjoying it.
Yeah, that's great. So another question that I love asking people is, if you were not a scientist, what would you be? What would you do? Have you ever thought of that?
Certainly thought about it. I think probably in a practical sense, I come from a family of lawyers, and it's actually a career track that I considered after graduate school, something that I considered, in the middle of my postdoc. I don't think I would have been very good at it, but might have been a practical option. But in terms of things that would be more fun, I've thought about food trucks. I don't know, something about operating a mobile kitchen that has always drawn to me, and some of my favorite food is from food trucks. And so, yeah, when I was a boy, I wanted to be a pizza delivery driver. So you can tell that I'm drawn to food.
Amazing.
Yeah.
Do you like to cook?
Do I like? So after just saying that, I would operate a food truck.
Or would you have to hire a cook?
Yeah, I
For
would
to
probably
sell
have
things.
to. I would say I'm very privileged to have a very talented wife who cooks very well, and she, explores different cuisines, and so I've definitely benefited from that. I really like to make sushi.
Nice.
Something we do on a pretty regular basis, and so, but that might be the extent of my culinary skills.
Well, that's pretty difficult. I can never get the rolls to stay together. So
The trick is in the rice, having really good rice. Yeah.
Thanks for that tip. Gonna try it again now. And the last thing I wanted to ask you is we always ask our guests for a little piece of advice. And I think with your experience in cryo-EM training, is there any piece of advice that you would give students who are interested in a career that involves cryo-EM?
I would say the first thing that comes to mind with that question is something that I tell everyone who's interested in joining my lab, is that, as long as they're surrounded in this environment, is to ask questions. In my experience, that's always been the fastest way to learn. And I would say it's even more important so now, because my sense is that, we have moved into a more isolated mindset, naturally, because of the pandemic and emerging out of the pandemic, where we've gotten used to isolation and gotten used to trying to figure things out on our own. And I think to an extent, we have to do that. We have to do our own due diligence. But for trainees out there who have the benefit of being surrounded by expertise, ask questions. I love it when people can just walk into my office and just want to chat. I love it when people drop me cold call emails, after browsing cryo-EM 101 and wanting to learn more. And I always make it a point to respond to cold call emails. And so that's the first thing that comes to mind, is to ask questions, because it is the fastest way to learn.
That's great advice. And I think that's also a good thing to close on, after I asked you so many questions and you've encouraged people to ask more questions. I think that brings us to the end of our episode today. So thank you very much, Peter, for being our guest today. And thanks everyone else for listening to or watching cryotalk.
Thanks, Eva.